My presentation is on how the past climate shapes affected the genetic diversity over Africa using an idealized modeling study. We used the agent-based modeling and the beast model for the phylogenetic analysis. The climate shifts played a very important role in the past human migration and possibly the genetic diversity it created, and the orbital scale variability, the Milankovitch cycle including the obliquity, eccentricity and precision had played a role in changing the rainfall pattern and the vegetation pattern over the Africa, and this could finally eventually change the habitat suitability of early humans over the African regions. The interplay between the environmental changes, human migration, adaptation and the population dynamics created Africa a very genetically diverse region. This figure shows the genetic diversity over the Africa, the major mitochondrial haploid groups over the Africa. You can see the L0 to L6. The different regions of Africa is dominated by different haploid groups. You see in the south it is L0 and the west it is L2, L3, they are dominating. Coming to the habitat suitability of the Heidelbergensis taken from Timberman et al. 2022. This figure shows the habitat suitability. The blue shading represents the habitat suitability of Heidelbergensis, and these dots, these boxes shows the Heidelbergensis sites. It is the evidence are showing that around 850 to 650 kilo years ago, this Heidelbergensis split into two groups. One is in the north and one is on the southern group. This will be probably due to the high eccentricity over this period 680 and 580 kilo years. These insets shows the habitat time variation of the habitat suitability over the past 800,000 years. You can see habitat suitability were very high over around 680 and 580 due to the eccentricity modulated influence. During this time period, the green corridors were opened and the humans migrated into the different parts. The human branch, the Heidelbergensis branch that migrated to the south eventually turned into the Homo sapiens, and those who went to the Central Africa finally changed into the Homo neanderthals around 4,000 to 5,000, 400 to 300 kilo years ago. So coming to the more recent period, the habitat suitability of Homo sapiens. This figure shows the net primary productivity and the habitat suitability variability in the last 200,000 years. You can see the net primary productivity changes with the orbital cycle, and according to that the habitat suitability also varying. You can see the greening of Sahara and the desertification of Sahara during the course of time. This influenced this helped to create green corridors between the regions where they are not connected earlier, and the opening and closing of the green corridors affected the human migration over these regions. Humans will migrate and it will also affect the cultural exchange between the populations. One example is the ostrich eggshell bead transfer between the East and South African region. This ostrich eggshell bead technology is probably evolved in the eastern part of Africa around 50,000 years before, and 50 to 33,000 years ago it was transferred to the southern part of Africa, and at the 33,000 years ago the ITCZ shifted downward. It affected the rainfall pattern, and it created more flooding events of the Zambezi river catchment that is nearly here. So it blocked the movement of people from the north to the south. So after some time, actually up to the 13 Ka, this ostrich eggshell bead custom was merely absent over the southern part of Africa, and it re-established after 19K, and these two things merged, population merged around 2000 Ka, and these customs re-established over these regions. So, this motivated us to study how these past climate change, past climate shifts change the human migration and the genetic diversity over Africa. We used the agent-based model and the beast bioinformatic tool for these studies. The agent-based model is used for simulating the movement of the agents, and the output of the agent-based model is given to the beast model for the phylogenetic analysis. Here, this beast is for the molecular sequencing. By it will use the Monte Carlo chain method for the sampling, and it used the time rooted, time measured phylogenetic information analysis of these informations. So, coming to the schematic, this is a workflow. The left hand side is the agent based model, and the right hand side is the beast model. We will simulate the agents with the agent based model, and then the output is selected agents will, the DNA information of selected people will be given to their beast system. The input of the agent based model includes net prime NPP and the habitat suitability. This MPP is calculated from this 2 million year CSM simulations conducted at ICCP, and we use the temperature and precipitation and tree fraction to evaluate the NPP, and with the NPP and fossil and archaeological records we generate the habitat suitability. It is given to the agent based model, and the output is given to the beast. Here we use a BT software which is for preparing the input file for the beast, and the output of the beast, mainly the tree files, will be analyzed using fig tree tracer and tree annotator. Coming to the agent based simulations, here we used an ensemble of idealist simulations by allowing time varying climate and fixed climate. In the time varying climate, the net primary productivity and the habitat suitability will change over time. We did the simulation for the past 200,000 years, and the other set of experiments are the fixed climate experiments. One is the we fix the climate of the pre-industrial time, and the experiment is run for 200,000 years, that is called a fixed sky 0K, and the other experiment we fixed the climate of 184K and did the simulation for the 200 for the time period. Here we didn't account for the cultural evolution, only just migration, and we track the mitochondrial DNA of each individual. The specification of the model is shown in this table. The maximum age is 55, and the fertility age is different for male and female. It is for male it is 13, for female it is 11, and the maximum carrying capacity is 35, and our carrying capacity will change according to the net primary productivity and the habitat over the regions. The mutation rate is 2.3 in 10 raised to -8 sites per year, and we conducted an ensemble simulation. With the climate change, we created 20 ensemble members, and the other two simulation it is 15 ensemble members. This figure shows the number of people and the precision cycle. The green line shows the precision cycle, and the other shows the number of people over the past 200,000 years. Can see in the varying climate simulation, the number of people is following the precision cycle, it is changing according to precision cycle, it is going up and down. But for the other fixed climate simulation, since climate is not changing, this remains nearly constant. Now, why we choose the present time and this 184K, because their number of people are maximum and minimum. Coming to the spatial distribution of humans in the past 200,000 years, we first figure shows the varying climate. Here first we introduce the people over the eastern part of Africa and just allow it to migrate and evolve according to the time. Here we can see the agents are moving north and south and different regions according to the habitat suitability of the region. And if we come to a fixed climate, the habitat suitability is kept constant, so human distribution remain relatively constant. See this. And if you see the two fixed climate simulations, the pattern is different because of the habitat suitability is different. In this fixed 0K, these two regions are not connected, but here it is connected. And if you see here, there is connectivity is very poor because of break over this region. So these simulations are different, and the migration of people are also different. We calculated the genetic distance, Nakamura genetic distance from all the experiments, and the dark line shows the mean, and the spread is also represented in this. The genetic distance is higher in the fixed climate simulations comparing to the varying climate simulation, and the fixed climate simulation is slightly higher than the earlier and the varying climate simulations. This low genetic distance in the varying climate may be due to the formation of green corridors over the time, so it will allow well mixing of the isolated populations. This figure shows the formation and disappearance of green corridors. This is a net primary productivity and habitat suitability for 5,000 kilo years before and 25,000 kilo years before. 5,000 kilo years before, in this ellipse region, the more vegetation is seen and the habitat suitability is higher over this region. So these two regions are well connected. But in 25 kilo years ago, these NPP values are relatively low, and the habitat suitability is also very low. So this will not allow for the free movement of agents or humans between these two regions. Coming to the beast simulations, here we use the beast version 2.6.7, and for the phylogenetic analysis of mitochondrial DNA of each people. In the agent-based model, we track the mitochondrial DNA also. So with that, we generated the DNA information of the people. The first person's DNA is generated by using a random sequence generator in MATLAB with 17,000 base pairs, and we selected every 100 people at every 4,000 years and given this the information to the beast model for the phylogenetic analysis. Here we allowed the basic base exchange at between A and G and C and T only for our experiments. We use these are the parameters used for the beast simulation. The substitution model is GTR. We use a strict clock, and a coalescent base skyline is used for the tree prior. The chain length is 10 million, and the clock rate is same as the agent-based modeling. The mutation rate is same, 2.3 × 10⁻⁸. And this output of the beast simulation is the tree is consolidated by using the tree annotator by leaving first 20% of the tree. This figure shows the consolidated tree from the three simulations with the equal branch length. The branch length is kept as unit for every branch length. This shows the first one is the varying climate and other two are the fixed climate. From the varying climate simulations, these number of branches are relatively higher. You can see it is higher than 70, most of the branches. It is compared to the other two simulations, it is relatively higher, and it is the lowest in the fixed climate simulation. The number of branch lengths are very, length of the branch lengths are very, number of branches are very less in this simulation. You can see clearly in this simulation, in this plot, shows the probability density and the branch length for the three sets of experiments. The probability density peaks around 2,500 year branch length, and it is slightly higher for the varying climate simulations, and for the higher branch lengths, more than 10,000 year, it is higher for the fixed climate simulations. So in the fixed climate simulations, the branch length are generally higher than the control simulations. This is a summary. We did two types of agent-based modeling experiments. One is with climate change, and other one is a fixed climate change. These both, the fading climate change simulated well the human distribution, and the fixed climate also simulated really a stagnant pattern, and the genetic diversity is marginally greater in the fixed climate simulations compared to other simulations, and the tree diagram also shows significant difference between the simulations. So the references. Thank you.